Hello friends, welcome back to the lecture. So I will be discussing today about the physiology of insulin and ketosis. This is a very basic lecture and this is intended to refresh the basics. So if you already know the basics, you can skip this lecture. I'm Dr. Santosh Abraham. I'm working as a specialist registrar in endocrinology and diabetes at Scarborough General Hospital. So the topics covered in this lecture will be about insulin, mostly its structure, synthesis, mechanism of action, and insulin secretion, regulation, glucose transporters, then ketone body formation and metabolism, and finally ketosis. So introduction and history of insulin. The word insulin is derived from the Latin word insula. It means island. In 1869, Langerhans identified the alpha and the beta cells in islets of pancreas. In 1889, von Mering and Minkowski produced experimental diabetes. Banting and West in 1922 extracted insulin from pancreas. And insulin boasts itself that it was the first hormone to be isolated in a pure form, and it was the first protein in which the complete amino acid sequencing was done, and it is the first protein produced by recombinant DNA technology. The structure of insulin. Insulin is a protein hormone with two polypeptide chains. The alpha chain has 21 amino acids and the beta chain has 30 amino acids. These two chains are joined together by two disulfide bonds. They are two disulfide chains actually, and these are linked between A7 and B7 and A20 and B19. In the A chain there is also an intrachain disulfide link. Synthesis. It is synthesized in the rough endoplasmic reticulum of the B cells, and then it is transported to the Golgi apparatus, where it is packaged into membrane-bound granules. These granules move into the plasma membrane and they are expelled by exocytosis. The insulin then crosses the basal lamina of the base cell and a neighboring capillary, and it finally reaches the bloodstream. The gene is located on the short arm of 11, chromosome 11 in humans. It has two introns and 3 exons. Pre-proinsulin is formed first, and it originates from the ER. Then the remainder of the molecule is folded and disulfide bonds are formed to make the proinsulin. The peptide segment which connects the A and B chains is called the connecting peptide, or the C peptide, and it facilitates folding. Then it is detached in the granules before secretion. There are two proteases involved in processing this proinsulin. Normally 90 to 97% of the product released from B cells is insulin, along with equimolar amounts of C peptide. C peptide can be measured by radioimmunoassay, and its level in the blood provides an index of B cell function in patients receiving exogenous insulin. In short, it is a protein synthesized and secreted by the beta cells of the islets of Langerhans. It's synthesized as a larger precursor polypeptide chain called proinsulin, which has 109 amino acids, rapidly converted to proinsulin in the ER by removal of 23 amino acids. The proinsulin has 86 amino acids, and it is transported in the Golgi apparatus, where it is cleaved by a protease. The C peptide with 33 amino acids is removed, and insulin with 51 amino acids is thus formed. See, here you can see the proinsulin and then the active insulin. which has got the two disulfide chains connecting the two subunits, and an intra disulfide chain in the A subunit. Metabolism of insulin. Half-life is around 5 minutes. It binds to insulin receptors and some is internalized, destroyed by proteases in endosomes formed by endocytosis. Factors increasing insulin secretion. These are the following. One is glucose, about which we will discuss in the next slide. Then GI hormones like secretin, pancreasymin and gastrin, proteins and amino acids, parasympathetic and beta adrenergic stimulation, glucagon and growth hormone, drugs like sulfonylurea acid or butamide, incretin hormones like GIP, GLP-1. This is a slide which shows the glucose entry when and the subsequent release of insulin from the granules. So when glucose enters the beta cell through GLUT2 transporter, it activates the potassium ATP channel. There is increased cytosolic entry of calcium, which will stimulate the beta cells, and insulin stored in granules is secreted outside. So the effects of glucose on insulin secretion. We all know that glucose is a major stimulant, and as the blood glucose level increases, insulin secretion also increases. Glucose induces a biphasic response to insulin secretion. A discharge of the insulin from beta cell occurs during the initial rapid phase of insulin release within first 2 minutes. The second phase of insulin lasts for 5 to 10 minutes and is of smaller magnitude and is due to discharge of newly synthesized hormone. As I've shown you before, the beta cells have GLUT2 receptors through which glucose is absorbed. Glucose is oxidized and more ATP is produced. ATP stimulates the ATP binding cassette protein, which is also referred to as the sulfonylurea receptor. Potassium channels are closed and calcium channels are opened. Increased intracellular calcium causes the insulin secretion. Factors which decrease the insulin secretion are epinephrine and alpha adrenergic stimulation. So you can see in this slide the stimulators and inhibitors: glucose, mannose, amino acids like leucine, arginine, GIP, GLP-1, beta keto acids, acetylcholine, glucagon, cyclic AMP, beta adrenergic stimulators, theophylline, sulfonylureas, all stimulate insulin secretion. So this could be one of the reasons why beta adrenergics and theophylline could produce hyperglycemia. On the right hand side, you can see the inhibitors like somatostatin, T2DG, manoheptalose, alpha adrenergic stimulation, beta adrenergic blockers, diazoxide, thiazides, hypokalemia, phenytoin, alloxan. You can infer from this slide that propranolol, thiazides, and alpha adrenergic stimulation all can cause hypoglycemia due to the inhibition of insulin release. I wanted to highlight cyclic AMP and insulin secretion in this slide because stimuli that increase cyclic AMP levels in beta cells increase insulin secretion. These include beta adrenergic agonists like salbutamol, glucagon, and phosphodiesterase inhibitors like theophylline. Catecholamines have a dual effect. They inhibit insulin secretion via alpha-2 adrenergic receptors and stimulate insulin secretion via beta-adrenergic receptors. The net effect of catecholamines is usually inhibition, but if they are infused after alpha blockade, the inhibition is converted to stimulation. The thiazides, which cause hypokalemia and which cause loss of potassium and sodium, decrease glucose tolerance and makes diabetes worse. This is primarily thought to be because of the K-positive depleting effects. So if in such patients, in diabetes patients who have bad blood sugar control, potassium sparing diuretics such as amiloride could be substituted. Mechanism of action of insulin. I will describe this under the following headings. First is the binding of the insulin with the insulin receptor, which is then followed by signal transduction and then gene transcription, which leads to activation of enzymes, and also DNA synthesis and glucose uptake. Insulin, when it binds to its receptor, which is on the plasma membrane, is the first step. In obesity, the number of receptors are decreased and target tissue becomes less sensitive to insulin. So this receptor is a glycoprotein. It has 4 subunits, 2 alpha and 2 beta subunits. The alpha subunits are located on the extracellular side, and here the insulin binds. The beta subunits are exposed on the cytoplasmic side, and the beta subunit has tyrosine kinase activity. When insulin binds to the alpha subunit, it activates the tyrosine kinase activity of the beta subunit, and it leads to autophosphorylation of the beta subunit. This phosphorylates insulin receptor substrates, and the message is later transmitted into a series of serine threonine kinases such as IRS, RAS, RAF, and MAPK, which will cause a cellular response. This slide thus summarizes the signal transduction. Insulin acts at the transcription level also to regulate synthesis of more than 100 proteins. The clinical significance is that it induces the following enzymes: glucokinase, pyruvate kinase, phosphofructokinase, and acetyl coenzyme carboxylase, those which are involved in glycogenesis and lipogenesis. And it will repress the following enzymes: glucose 6-phosphatase, PEP carboxykinase, and fructose 1,6-phosphatase, which are involved in lipolysis and gluconeogenesis. So insulin induces glucokinase, GCK, pyruvate kinase, PK, PFK, and acetyl coenzyme carboxylase, and it represses glucose 6-phosphatase, fructose 1,6-biphosphatase, and phosphoenol pyruvate carboxykinase. Insulin also activates enzymes by covalent modification, phosphorylation or dephosphorylation. There are more than 50 enzymes activated by this mechanism. So it activates protein phosphatase I, which dephosphorylates enzyme proteins. The enzymes which are activated are glycogen synthase, pyruvate dehydrogenase, pyruvate kinase, acetyl coenzyme carboxylase, and HMG CoA reductase. These are the enzymes whose activity is increased. DNA synthesis. Through the IRS1 pathway, it increases DNA synthesis, cell growth, and anabolism. The intracellular mediators in this role are calcium and cyclic AMP. Insulin activates phosphodiesterase and decreases cyclic AMP. So reactions dependent on the cyclic AMP are inhibited. Finally, it increases the glucose uptake by recruitment of GLUT4 in cells. What are the physiological actions of insulin? Here we are going to discuss about the uptake of glucose, the utilization of glucose, hypoglycemia, lipogenesis, anti-lipolysis, anti-ketogenesis, and other effects. You can see that there are three different types of actions of insulin. One is rapid, the other one is intermediate, and the third one is delayed. So the rapid action results in increased transport of glucose, amino acids, and potassium into insulin sensitive cells. The intermediate action results in stimulation of protein synthesis, inhibition of protein degradation, activation of glycolytic enzymes and glycogen synthase, and inhibition of phosphorylase and gluconeogenesis. The delayed action is increase in mRNAs for lipogenesis and other enzymes. Uptake of glucose by tissues. It facilitates the membrane transport of glucose. The facilitated diffusion of glucose in muscle is also enhanced by insulin. In diabetes mellitus, GLUT4 is reduced. However, glucose uptake in liver is independent of insulin. Utilization of the glucose. Glycolysis is stimulated by insulin. You know, glucokinase, fructokinase, and pyruvate kinase activities are enhanced. Glycogen synthase enzyme is activated, so it favors glucose storage as glycogen, and also insulin favors fatty acid synthesis from glucose, and hence glucose utilization is also increased. Hypoglycemia. It lowers the blood glucose level by promoting utilization and storage. Gluconeogenesis is inhibited by repressing the key enzymes, pyruvate carboxylase, phosphoenol pyruvate carboxykinase, and glucose 6-phosphatase. Insulin also inhibits glycogenolysis by favoring inactivation of glycogen 6-phosphorylase and inhibiting glucose 6-phosphatase. By the net effect of all these three mechanisms, blood glucose level is lowered. Lipogenesis. It is favored by providing more acetyl coenzyme by pyruvate dehydrogenase reaction. It increases the activity of acetyl coenzyme carboxylase, and it provides glycerol for esterification of fatty acids to TAG. Insulin also provides NADPH by increasing the GPD activity of the HMP shunt. Anti-lipolysis. It inhibits lipolysis in adipose tissue by inhibiting hormone-sensitive lipase. The increased level of free fatty acids in plasma in diabetes is due to the loss of this inhibitory effect on lipolysis. Anti-ketogenesis. It depresses the HMG-CoA synthase, so ketogenesis is decreased. Also, acetyl coenzyme is completely utilized in the citric acid cycle because oxaloacetate is plenty. So insulin favors fatty acid synthesis from acetyl coenzyme also. So the acetyl coenzyme is decreased. All these factors thus reduce the availability of acetyl coenzyme, and hence the production of ketone bodies are reduced. Other general effects: protein synthesis is promoted and degradation is retarded. It is an anabolic hormone. It stimulates replication of cells. It is an essential growth factor for all mammalian cells. Now coming to glucose transporters. We all know glucose enters either by facilitated transfusion or by secondary active transport with sodium in intestine and the kidneys. In muscle, adipose tissue and some other tissues, insulin stimulates glucose entry into cells by increasing the number of GLUTs in the cell membrane. The GLUTs that are responsible for facilitated diffusion of glucose across cell membranes are a family of proteins. They span the cell membrane 12 times and they have the amino and carboxy terminals inside the cell. They differ from SGLTs in that they don't have any homology with them. SGLTs are responsible for the secondary active transport of glucose in intestine and renal tubules. Although SGLTs also have 12 transmembrane domains like the GLUTs. So what is the influence of insulin on GLUT? In the insulin sensitive cells, there is a pool of GLUT4 molecules inside the vesicles of their cytoplasm. When insulin receptors are activated, the vesicles move rapidly into the cell membrane and fuse with it. They insert these transporters into the cell membrane. When insulin action stops, the transporter containing patches of membranes are endocytosed and the vesicles are ready for the next exposure to insulin. This is how insulin via GLUT4 helps to transport glucose. Activation of this insulin receptor brings about the movement of vesicles to the cell membrane by activating phosphatidylinositol 3 kinase. Other glucose transporters are not insulin sensitive. Most of them are not insulin sensitive and they appear to be constitutively expressed in cell membrane. So independent of the GLUT4 transporters, insulin increases the entry of glucose into liver cells. It does this by inducing glucokinase, and this increases the phosphorylation of glucose, and hence the intracellular free glucose concentration stays low. So it facilitates the more entry of glucose into the cell. In exercise, insulin sensitive tissues which contain a population of GLUT4 vesicles move more into the cell membrane. So exercise induces this movement of GLUT4 vesicles into the cell membrane, and this is independent of the action of insulin. This is why exercise lowers blood sugar. So these are the various glucose transporters in mammals. So SGLT1 is found in small intestine and renal tubules, and SGLT2 is found in the renal tubules as well. By facilitated diffusion, so SGLT1 and 2 is responsible for the secondary active transport, that is a sodium glucose co-transport, while the glucose transporters are responsible for the facilitated diffusion. Especially the GLUT4 is stimulated by insulin. It's found in skeletal, cardiac, adipose tissue and other tissues. GLUT2 is the beta cell glucose sensor. So it's found in beta cells of islets, liver, epithelial cells, kidneys, etc. GLUT1 and GLUT3 are responsible for the basal glucose uptake. So it's found in brain, placenta and kidneys. Also, GLUT1 is present in red cells. GLUT5 is responsible for fructose transport, and that is found in the sperm as well. Let's come to metabolism of ketone bodies. The acetyl coenzyme A, formed from fatty acids, can enter and get oxidized in the tricarboxylic acid cycle only when carbohydrates are available. So during starvation and diabetes, the acetyl coenzyme A takes the alternate fate of formation of ketone bodies. Ketogenesis. So this is the formation of ketone bodies. Acetoacetate is the primary ketone body, while beta hydroxybutyrate and acetone are secondary ketone bodies. And these are synthesized exclusively by the liver mitochondria. So these are the steps in ketone body formation. First step is condensation, which involves two molecules of acetyl coenzyme A to condense to form acetoacetyl coenzyme A. The second step is production of HMG-CoA, so that is beta hydroxymethyl beta methyl luteal coenzyme. So one more acetyl coenzyme is added to acetoacetyl coenzyme A to form the HMG-CoA. It's interesting to note that the mitochondrial HMG-CoA is used for ketogenesis, while the cytosolic fraction is used for cholesterol synthesis. Lysis. The HMG-CoA is lysed to form acetoacetate. Acetoacetate is also formed by degradation of carbon skeleton of ketogenic amino acids like leucine, lysine, etc. HMG-CoA lyase is present only in liver. So this takes place in the liver. Reduction. Beta hydroxybutyrate is formed by reduction from acetoacetate. This ratio between acetoacetate and beta hydroxybutyrate is decided by the cellular NAD/NADH ratio. And acetone is finally formed by spontaneous decarboxylation from acetoacetate. Ketolysis. The ketone bodies are exclusively formed in the liver, but they are utilized by extrahepatic tissues. The heart muscle and renal cortex prefer ketone bodies to glucose as well. So brain and skeletal muscle also utilize ketone bodies if glucose is not available. Acetoacetate is activated to acetoacetyl coenzyme A by thiophorase enzyme. The acetoacetyl coenzyme A then enters the beta oxidation pathway to produce energy. So that is how acetoacetate is utilized in these extrahepatic tissues with the help of the thiophorase enzyme. Ketosis. So the rate of synthesis of ketone bodies by liver is such that they can be easily metabolized by the extrahepatic tissues. So the blood level of ketone bodies is less than 1 milligram per deciliter on any occasion, and only traces are excreted in the urine. But when the rate of synthesis exceeds the ability of the extrahepatic tissues to utilize them, there will be accumulation of the ketone bodies in blood. This leads to ketonemia, ketonuria, and smell of acetone in breath. And all these three constitute the condition called ketosis. So causes of ketosis. We all know diabetes mellitus causes that. So it is the most common cause. Even though glucose is in plenty, deficiency of insulin causes accelerated lipolysis and more fatty acids are released into the circulation. And these fatty acids are oxidized and they increase the acetyl coenzyme A pool. Also, enhanced gluconeogenesis restricts the oxidation of acetyl coenzyme A by TCA cycle, since oxaloacetate is less. Starvation is another cause of ketosis. So that dietary supply of glucose is decreased. Available oxaloacetate is channeled to gluconeogenesis. The increased rate of lipolysis to provide the alternate source of fuel is thus enhanced. The excess acetyl coenzyme A is converted to ketone bodies. The higher glucagon favors ketogenesis, and brain derives 60 to 70% of energy from ketone bodies under starvation. Explanation behind ketogenesis. In starvation and diabetes, the level of glucagon is increased. Glucagon inhibits glycolysis. It activates gluconeogenesis, it activates lipolysis, and moreover it decreases the malonyl coenzyme A level, and thus it stimulates ketogenesis. A high glucagon insulin ratio is potentially ketogenic. Regulation of the ketone body formation occurs at the following three levels. The first level is lipolysis. So free fatty acids are the precursors of ketone bodies. So the factors regulating the mobilization of fatty acids from adipose tissue will control ketogenesis. Insulin inhibits lipolysis, while glucagon favors the lipolysis. The second level is the entry of fatty acid into mitochondria. The mobilized fatty acids now enter the mitochondria for beta oxidation. Carnitine acyl transferase CAT1 regulates this entry. Malonyl coenzyme A is the major regulator of CAT1 activity. In diabetes and starvation, glucagon increases and it decreases malonyl coenzyme A. So beta oxidation is stimulated. Oxidation of acetyl coenzyme A. This is the third level. When the above two steps are increased, more acetyl coenzyme is produced. Usually acetyl coenzyme A is completely utilized in the TCA, but in diabetes and starvation, glucagon insulin ratio is increased and key gluconeogenesis enzymes are activated. Oxaloacetate is diverted for gluconeogenesis, so citric acid cycle cannot function optimally. Thus, on one hand, acetyl coenzyme is generated in excess. On the other hand, its utilization is reduced. Thus, excess acetyl coenzyme A is channeled into the ketogenic pathway. In both diabetes and starvation, oxaloacetate is channeled into gluconeogenesis. So, the availability of the oxaloacetate is decreased. Hence, acetyl coenzyme A cannot be utilized in the TCA cycle. I want to just discuss about the regulation of beta oxidation here because it is related with the ketosis. So FFAs regulates the net utilization through beta oxidation. The level of FFA is controlled by glucagon insulin ratio. Glucagon increases FFA and insulin has the opposite effect. CAT1 is the regulator of the entry of fatty acid into mitochondria. Malonyl coenzyme A inhibits CAT1. Thus, during de novo synthesis of fatty acid, beta oxidation is inhibited. Thank you for watching this presentation. I hope this was helpful.